Hi, everyone. My name is Emma Zeitler, and I am a master's student in the University of Florida's Deer Lab under the advisory of Dr. Marcus Lashley. And so while we're up at Tall Timbers, I'll be leading y'all through some experiments that are relevant to my master's research project, but they'll also shed some light on the importance of wildlife interactions and how management strategies can affect them. So at the center of my project are white-tailed deer. And before starting this research, I'd mainly heard about white-tailed deer in the context of overpopulation or how they have negative impacts on habitats, specifically how they greatly depreciate plant biomass. And that's sort of the dominant narrative around this species. But we kind of try to approach our research considering deer as a potential ecosystem asset that can be channeled using management strategies. And so to begin, white-tailed deer are found pretty ubiquitously throughout North America, and they're known to amass large populations and travel long distances. And in many of these places, specifically Florida's longleaf pine ecosystems, they are the largest remaining herbivores because we've extirpated all the others. And so they often fulfill the ecological processes associated with this role. And in this way, they can be really important and impactful in their ecosystems. And so as herbivores, deer love to eat, and they will eat a lot. And when fruit is available on the landscape, that will be their preferred forage. And this is especially true in the fall when fruit is most available and deer are going through their lactation period, so nutrient demand is at its peak. So they'll be eating a bunch of fruit, digesting it all, and that brings us to our process of interest, which is deer-mediated seed dispersal. And so many plants in the LLPE have adapted to take advantage of animal consumption by producing fruit and seeds that can survive the animal digestive system. These seeds are then deposited on the landscape, wrapped in scat, and this provides them protection and nutrients, and then increases their chances of successful germination. And so deer have been shown to be major dispersal vectors with up to 11 species of plants found per scat pellet. And in this way, they can have a huge impact on genetic diversity, species ranges, and plant community composition. But of course, none of this happens in a vacuum, and this entire process is open to interactions with other plants and animals. And so one potential addition to the process is a secondary disperser. And this, the secondary disperser further disperses the seeds after the deer has defecated them onto the landscape. And so commonly seed dispersal is thought of as a single stage process, sort of solely just carried out by this primary role. But many plants have been known to benefit from a two-stage dispersal process. So just to separate these two roles out, the primary disperser can move seeds large distances, which is really good for enhancing genetic diversity. And they're also responsible for depositing the seeds in their scat package. But once on the landscape, these seeds are vulnerable to a variety of threats, such as desiccation or seed predators. And so this is when the secondary disperser can create an advantage by relocating the seeds to a favorable microsite, which often just means that they're being buried underground. And here the seeds are protected, they're resourced, they're in their forever home. And then additionally, the secondary disperser can break up the seeds away from each other, and this further reduces competition dynamics. And so this is a really important addition to the entire process. And the secondary disperser role is often fulfilled in the longleaf pine ecosystem by dung beetles. And dung beetles rely on animal scat for food and nesting purposes. So upon locating scat, many species will bury it underground, and they are then unintentionally secondarily dispersing these seeds. And bearing behavior by dung beetles has been shown to increase germination rates by up to threefold. So when considering management strategies, if you're looking to boost your forest health or plant community composition, this is obviously an interaction of interest. And then while I'm on my soapbox, just to talk a little bit more about the importance of dung beetles, they're actually really important in other services like nutrient subsidization and soil processes. And just in the US, they're estimated to contribute around $300 million in ecosystem services per year. And so they're often introduced to agricultural areas, or famously the entire continent of Australia, as a management strategy. So they're generally just an important organism to pay attention to. And so one land use we've been thinking about in this context is hunting. And so without hunting pressure, deer and dung beetles are both active during the day. And there are a few exceptions with the dung beetles, as some of them are crepuscular or nocturnal, but the majority are likely diurnal, and this allows them greater access to the animals that they're dependent on. However, when hunting pressure is introduced, deer are known to shift their activity habits so that they peak at night. And this mismatches dung beetles from their life source, and it mismatches seeds from their secondary disperser. And so hunting is allowed on the Tall Timbers property. And my own trail cameras that were active just this past hunting season showed that most deer activity did occur during the night. And so this brings us to the question, how does nocturnal deer activity affect secondary dispersal by dung beetles? And so to answer this question, we've designed a 2x2 factorial experiment, which means we'll have two different treatments and we'll investigate every combination of those treatments. And the basic concept of this experiment is that we're going into the longleaf pine habitat, which I've illustrated here with these two green blocks, and we're depositing pre-collected deer scat and then monitoring the scat for beetle activity. And the first treatment we're imposing is the time of scat deposition. So by us going out and depositing the scat either during the day or at night, we will be simulating deer activity habits. And this will allow us to see how dung beetle activity changes between those two periods. The second treatment we're using is a part of another investigation that asks if deer diet influences dung beetle preference and activity. So we will be using scat that has been collected either from deer that are eating a fruit-based diet, and we call this fruity poop, or deer that are eating an alfalfa-based diet, which is our control treatment. And we will be placing both scats down at the same time and running these trials simultaneously. And so this will tell us if one scat composition attracts more dung beetles than the other. And so each trial will begin when a group places a known amount of scat in the habitat. And this scat, it will be monitored by a game camera for about 12 hours. And at the end of the trial, the remaining scat will be counted, the SD cards will be collected, and then I'll further analyze the footage. And so measurements I'm going to take from the footage include the time of the day or night that the first scat was removed, how much time it took for that first scat to be removed, and then I'll also just verify the amount of scat that ended up getting removed. And so the hypotheses that we're testing, first A, we have that scat deposited during the day will be located and removed faster and more often than scat deposited at night. And then We'll also be testing if fruity scat gets removed faster and more often than the control scat. And so both of these processes have the potential to impact white-tailed deer's role in their ecosystems pertaining to their seed dispersal processes. And depending on what we'll find, our results will further inform how management strategies and wildlife phenologies interact to affect basic ecosystem processes. And so this might seem like a lot of new and weird information all at once, but it will definitely start making more sense once we're out there and doing the experiments. Until then, I included my email in the title slide. So if you have any questions, just feel free to send me an email. And otherwise, I will see you all out there.